Okay, so welcome to another screencast. Today we're going to be talking about membrane structure and function, but really what we're going to be focusing on is osmosis and diffusion and osmosis. So I'm going to go ahead and share my iPad so that we can see the PowerPoint slides. Okay. So hopefully we're going to have some time at some point to watch this, but I will also post a link because it's freaking cool. So we're going to be talking here, before we get into kind of cellular transport and how that occurs, we are going to focus on diffusion and osmosis. So we already talked about the fluid mosaic model, how membranes, I don't know if we're going to talk about this yet. This might be the first introduction to the fluid mosaic model, depending on what order I go. So the fluid mosaic model is a model that describes the plasma membrane, right? Describes the plasma membrane, where you have a membrane that is fluid. This is very, very important. And remember, when we look at that fluidity, we are talking about the phospholipids, right? How they are moving around. So for example, here's some phospholipids, and they are not static. They're highly mobile. Remember that the membrane is composed of phospholipids, which are amphipathic, meaning they have a polar head, right? And we have a core region of nonpolar tails. Great. The amphipathic phospholipids create what is called a semi-permeable barrier, where some substances can go through it and some can't. Right? So some substances can go right through that plasma membrane and others cannot. So if we look at this diagram here, small uncharged molecules such as gases and steroids, which are indicated here by these blue region. So these are gases and steroids. They can go directly through the plasma membrane, diffuse directly through, right? The gases that we're going to be talking about in this class, I think there's only 3 that we really talk about. We talk about oxygen, carbon dioxide, and at some point, we're also going to be talking about nitric oxide, right? And remember steroids, which are lipids, right? With those 4 carbon rings, right? However, if you have molecules that are large or charged or polar, meaning they have partially charged, those are represented here by the orange and the green, right? Those cannot get through that non-polar core, right? Which kind of acts as a jail. Can't get through. No. Polar, charged, large, cannot get through. So what they use as a gatekeeper are proteins, right? So membrane proteins, or proteins that are embedded in the phospholipid bilayer, will help get these kinds of molecules across the membrane, right? So membrane proteins, which are shown here in this kind of purplish color, help get proteins across the plasma membrane that are either large, polar, charged, right? Those kinds of molecules need help to get across the plasma membrane because they can't diffuse right through like the small gases and the steroids. All right, so what is diffusion? So diffusion, by definition, is just the movement of molecules from an area of high concentration to an area of low concentration. So we're always going to be talking about going from high concentration down to low concentrations, right? That is diffusion. Sorry, I think I need to move my face so you can see that. High to low. So you guys all have experience of this. Or you can go do this experiment. Take a cup of water, helps if it's see-through cup, and then just put a drop of food coloring in. And what you can see is that that solute, the dye, will move away from its area of high concentration down towards where it's less concentrated. It will be diffusing outward until it reaches this state of equilibrium, where there's about the same amount of concentration of solute or dye everywhere dispersed throughout the water, right? In the absence of other forces, substances will always diffuse down their concentration gradient, going from an area of high concentration to an area of low concentration. One thing that I think is really cool that I never really thought about before I started teaching this stuff is that both the dye and the water are both diffusing down their concentration gradients until equilibrium. So if we look just at, so for example here down in A, we have a crystal of dye that's placed in water. That crystal of dye is going to be moving away from its high concentration into the water, which has very low concentration of dye. However, at the same time, the water is going to be moving in towards the dye from high concentration of water to low concentration of water. So both of them are diffusing at the same time, right? And that keeps going until you have an equilibrium or an equal distribution of molecules. One great example of diffusion that I always like to bring up is the diffusion of gases. And in particular, we're going to be talking about oxygen and carbon dioxide. So a cool thing about oxygen, or sorry, about oxygen, about diffusion. So first thing, what is diffusion? Going from a high concentration, moving down towards where that thing has a lower concentration, from an area of high to low concentration. Diffusion, by definition, requires no energy because the energy is kind of stored in that concentration gradient. So everybody, take a deep breath. Ready? 1, 2, 3. And hold your breath while I talk. Okay, so as you're holding your breath, you have a lot of oxygen that has come in from the outside environment, and you have a high amount of oxygen there in your lungs. That high concentration of oxygen is going to diffuse down its concentration gradient into the blood vessels, all right? You can release out. But at the bottom of your breath, hold again, all right? So now at the bottom of your breath, you have not very much oxygen, and in fact, your blood is deoxygenated, right? Meaning that you have a large amount of carbon dioxide now in your blood that is, when it reaches the lungs, it's going to diffuse down its concentration gradient into the lungs, and then that's going to cause you to breathe it out. All right? So what we're talking about in these lungs is we're looking deep in the lungs at these kind of structures called alveoli, little air sacs, right? And what's really neat about them is that they're only one cell, there's only one plasma membrane there thick. So we have a phospholipid bilayer here, and then in the blood, we have one other phospholipid bilayer. So oxygen and carbon dioxide are really only having to diffuse through 2 plasma membranes. So when we talk about diffusion, it's important to think or to know that each substance is moving down its concentration gradient independent of the other ones. So let's look at just this first part where we're looking at the diffusion of one single solute or molecule kind of in water. So here we have a molecule of dye and we have a membrane going across it. So here is the little membrane and the dye can go through that membrane. So what we'll see is that we have a net diffusion of those molecules of dye going away from their concentration where it was a high concentration to a lower concentration. So they're going to move down that concentration gradient. At some point, we will see that there is an equilibrium where the amount of dye on both sides is equal, right? Things don't stop moving at that point, but there is equal moving in both directions, right? And that's equilibrium. If we look down here at the second example, here we have two solutes, right? One on each side. So if we look only at one first, we're going to look only at the orange balls. So if we look at the orange balls, they're going to go down their concentration gradient from an area of high to low. At the same time, the purple balls are going to be going down their concentration gradient from an area of high to low. So what you'll see is that the orange is going in this direction, right? Ooh, I wonder if I can color code that. Ooh, yeah. So the orange is going to be going in this direction, and the purple is going to be going in this direction, right? So both of them are moving at the same time, but they're moving in opposite directions depending on their independent concentration gradients. All right, and then again, in both of these scenarios, they're going to move until equilibrium is reached, right? And equilibrium basically means the same concentration, that's my abbreviation for concentration, same concentration of solute on both sides of the membrane. And it doesn't mean things stop in motion, it just means there's equal moving now. All right, osmosis is diffusion, but in particular, it's diffusion of water, right? We're talking specifically the diffusion of water molecules, right? Remember, we're talking about these polar water molecules across a semi-permeable membrane. So semi-permeable just means that some things can go through it, some things can't, right? So if we look at our example here, we have what is called a U-tube. Whoops, I almost spelled it like the link. U-tube, all right? And these U-tube questions have stumped students for years. Well, let's just take it slow, one at a time. All right, so in this U-tube, we have two different concentrations of water that we put in this tube, and there is a selectively permeable membrane across it. This particular semi-permeable membrane allows water to pass through, but not the solute. And I have kind of a diagram of that in this middle one, where the water molecules can move back and forth, but these big green chunks cannot move. So they get stuck on whatever side that they are on, but the water can move freely. All right, so if we look here on the left side, here we have a low amount of solute and we have a higher amount of water. If we look at the right side, we can see that there's more solute and less water. We know the solute cannot pass through the semi-permeable barrier. So what's going to happen? We are only looking at the water in this case because the solutes can't go through. So what we're going to see is that water is going to move down its concentration gradient, right? So water moves from an area of high concentration on the left to a lower concentration on the right. And what that does is that causes the water levels to go up on the right side, right? So water is diffusing down its concentration gradient until equilibrium is reached. Solutes cannot move. Okay, so this, for example, is time point, let's say 0, right? We just set it up this way, and this is at a later time point, right? Some later time point. All right. So let's look at our friend, the slug. All right, so here's a beautiful, cute little slug. Actually, I hate slugs in my garden, but whatever. My husband went to UC Santa Cruz. He loves a banana slug. All right, so what happens? Hopefully none of you have done this, but what happens when you put salt on a slug? So here's our little slug. I'm going to put a little pile of salt on it, right, from my little salt shaker. My little salt shaker, and I have a little pile of salt on top. What happens, right? So slug, what happens now is that the slug actually shrivels up, and that is because there's a high salt on the outside. Salt is polar in charge. It's not going to go through plasma membranes, right? We know that water can, because of aquaporins. If we haven't learned that yet, we will, so don't worry. Water can go through the plasma membrane because of these aquaporins. So what we see is that water has a high concentration on the inside. Where we put that salt, there's very little water. So what we're going to see is that water moves out of the slug into the pile of salt, and the slug shrivels up and dies, right? So we see, stop X, we're going from the water is moving from a concentration, from a high concentration inside of the slug, and it's going to move down its concentration gradient to the lower concentration outside of the slug, right? An example that we're going to talk about quite a bit in this class are red blood cells, all right, floating around our blood. And we're going to look at red blood cells in salt water, in blood plasma, and in pure water, all right? So we have these 1, 2, 3. So here's our number 1 case, our number 2 case, and our number 3 case. So it turns out that blood plasma, kind of the fluid surrounding blood cells, has a solute concentration similar to the red blood cells. So that is called an isotonic solution, right? Isotonic solution. And what happens in that, because there's equal amounts of solutes and equal amounts of water in both, we are going to see diffusion of water going out of the cell, kind of at the same rate that the water is coming in the cell, right? So this is what happens in an isotonic solution. There's equal movement. However, if we now put red blood cells in a super salty environment, as I'm indicating by all these little black dots, right? If we put those red blood cells in a salty environment, then there is a lot more water inside the cell. So here we have a high concentration of water inside the cells, and it's going to move down its concentration gradient to a lower concentration outside of the cell, right? And you can see water is going to move out, and those cells are going to shrink up, right? This is an example of a hypertonic solution, right? And the way I remember that, remember I drew all those little dots? I always think of that as hyper. Hyper, hyper, hyper. That means lots and lots of solutes, right? Solution has lots of solutes. All right. The last example is called red blood cells in a hypotonic solution. So in a hypotonic solution, what that means is that, babe, you gotta leave me alone, please. Bex, please, I'm in the middle of a recording, you gotta go. Give me 5 minutes, okay? All right, so in a hypotonic solution, what you see is that there is less solutes on the outside. So what that means is actually that the cells have more salts or solutes on the inside. So what that means is that water is going to move down its concentration gradient from high inside to low outside. Water is going to rush in, and you will actually see cells burst, right? So it's really important in our blood that our blood plasma is not too salty, shrivel the blood cells, or not too watery, and the cells would burst. All right, so what is the difference between animals and plant cells? So first, let me repeat those solutions. So a hypotonic solution is a solution that has lower solute concentration than the cell. An isotonic means that they have equal solute on the outside and the inside of a cell. In hypertonic, remember my hyper situation over here, where we are going to have the outside of the cell be, oh my goodness, there's like a million things happening right now. Sorry, phone's ringing, Dax is in the way. So oh my gosh, the hypertonic solution is where you have a high concentration of solute outside. All right, so let's just think of what would happen here. So again, in a hypotonic solution, water is going to move in. In an isotonic solution, they're equal. In a hypertonic solution, water is going to move out. Each of these, water is moving down its concentration gradient, right? Yeah, you're right. If you're thinking, but what about the solutes? Why aren't they moving? They might be able to move depending on what kind of solutes they are. But we're really just talking about water here because we're talking about osmosis, the diffusion of water. Okay. One other thing I'd like to point out is that here, in this example here, we have a hypotonic solution. However, that means the cell itself is hypertonic to that solution. If we look over here, the cell is hypotonic compared to the hypertonic solution, right? So you kind of have to, when you're talking about these, you need to talk about either the solution or the cell. Generally, we're talking about the solutions. All right, let's talk about plant cells. So for plant cells, if you put them, and we have those three scenarios again, we have a hypotonic, an isotonic, and a hypertonic solution. If we put a plant cell in, let's talk about animals first. So here's animals on top, plants on the bottom. Recap, isotonic solutions. Plant cell or animal cells, equal diffusion of water in both directions. Plant cells, equal diffusion of water across the plant cell, no problem. That for a cell is called a flaccid cell, right? It's normal, it's fine. However, it's not super happy, right? So that means that you can get a plant leaf that is kind of like a little wilty and sad, but it's still alive, right? If we put an animal cell in a hypotonic solution, water is going to rush in and it's going to burst, right? So animal cells burst in a hypotonic solution. However, plant cells, that is their preference. They like to have lots of water, and that leads to a turgid plant cell, or in other words, a plant cell that is nice and real happy, right? When you see a leaf kind of, that is flaccid, and it probably needs more water. In a hypertonic solution here, animal cells, the water will rush out. In plant cells, the water is going to rush out. However, plant cells, what happens is that they go through what is called plasmolysis, where the cell membrane kind of pulls away from the cell wall, right? So the cell membrane shrinks and pulls away. And often that is unrecoverable, and that plant cell could die, right? If we go back to this hypotonic solution, the reason that it's not bursting like an animal cell is because of that cell wall. So cell wall prevents the cell from getting too big and bursting, because that cell wall is nice and rigid because of that cellulose. All right, and we'll stop there.